Okay, good morning. Can you hear me?
Yeah.
Okay, well, I'd like to thank Kenton, Luigi, Dave for having a wonderfully informative day yesterday. I'm looking forward to learning a lot more today. But most importantly, I'd like to thank you for the ingenuity, the tenacity, the creativity of tackling these large data sets. I think it's going to make an incredible impact in how we approach science, and I'm very grateful to your service. So in this lightning talk, this laundry list of little things that we'd like to be able to do with our data sets, I'm talking to you from the perspective of a transplant surgeon. So we know, for example, that the CDC has indicated that 1 in 3 individuals in 2050 will be obese. And if you look at this individual, you wouldn't call him obese, you'd call him overweight. But unfortunately, the risk factor of disease is the same in this person and somebody who is grandly obese. And what this means in terms of mathematics is that there are going to be approximately 100 million people who will be obese in 2050. And if you take the data sets that have been already generated, it means that about 23% will have some sort of kidney disease as a consequence. And when you look at 22%, that's about 22 million people in renal failure 2 decades later. And so as a transplant surgeon, you shudder when you look at this data and you think, what can we possibly do for our patients? And there are options, but they're not very good. For example, if we embark on trying to provide dialysis to all our patients, you would have to put 50 dialysis chairs in every gas station in America and run that 3 shifts a day in order to dialyze all the people. And then on top of that, you'd only have 80% or 20% live in 5 years. So some cynics would say, well, then you've solved your problem. They'll be dead in 5 years. But it's an ongoing problem. So the next wave of people will be coming in. We can transplant these patients. There is a problem with the long-term immunosuppression. The optimistic time frame in which a kidney is functioning after transplant is about 20 years. And now that people are becoming obese earlier, so we have children that are obese, we're transplanting in the 30s instead of in the 40s, 50s, and 60s. So now we have 50-year-olds coming back for their second transplant. And that leads to the fact that we have a critical donor organ shortage. There's no way we're going to find 22 million. We have 100,000 on the list right now. We have about 40,000 we do a year. You can do the math. There's no way we'll have 22 million organs. We can work on regeneration, similar to what a salamander might do to regrow a tail. We could try to regrow the kidney and help it regenerate. Both of these two, transplant and regeneration, are what I work on in my lab. Of course, the most important thing is prevention. And I did go back to school, get an MPH degree just to address this. But you know what? Our health services are not interested in this option. They're willing to fund the other two more. So our areas of study are span from mouse, monkey to human. And we take high-risk investigative science in mice and then translate it to monkey and then go to human. And so this is the picture of a glomerulus. This is what we're interested in when we're monitoring the tissues. The glomerulus and the tubules are the most important thing, but you can see that they're in different regions of the kidney, so that when we analyze these data, you can see that the glomeruli are to the outside in the cortex, and the tubules are on the inside, meaning that changes in the data have to be sort of designed before we can analyze this. And that means that I have to have a student spend several hours actually drawing these regions of the cortex and the glomeruli. And if you take the sections of the kidney, 5 different sections, and you have to draw 20 sections of 20 views and 5 sections per view, it's 100. You can see the math. This is days and days of work of drawing. And then the second problem is that if you generate this image, the middle image is an H&E image, and then the other two are different ways of analyzing the image, we get different software, different aspects of analysis, and we have to search for software that will help us. And last, if we have different types of images, this is confocal imaging, we have to use yet another software to be able to coalesce all the things. We hope to be able able to put these together in one total histologic understanding, but there's a lot of non-crosstalk between all these softwares. And the second thing, because he did ask me to talk about a second thing, so I get 1 more minute, so that was all on imaging. So the second thing is documents. And so there is in clinical trials this thing called Table 1. And if you look at it carefully, you can see that in Table 1, there are all the demographics that are required for that study that has been represented. So if you have a study of 10,000 people, you'll be able to look at this table and know exactly who was in that study. If one could sort of extract these tables so that as one is searching for data, this would be incredibly helpful in designing future studies and understanding where other studies have been. So in summary, the two sort of tool categories that we work with are imaging and documents. And in imaging, it would be wonderful if we could have an automated recognition of structure. That means have our softwares talk to each other so that when we draw medulla and we draw cortex and we have it in H&E or other different stains, they can all be conversant with one another. We don't have to switch from all different programs. Second, to be able to look at, and I kind of covered second, and then the third is to be able to extract similar images from the literature and be able to then import those meaningful data to our own data so that we can understand it better. And then in terms of documents, the ability to import a Table 1 for a cross-reference of all the data, and be able to sort of make searches based on genes where you can actually correlate gene pathology to gene clinical manifestation. So I'm sorry, I did 2, so I took a little more time, but those are the things. I'll write this down for you, Kenton. Okay. All right, that's it.
Maybe